Thanks for the introduction, and thanks for inviting me to give a presentation at this exciting symposium. I think it's nice that my talk is following the previous speaker because similar to the previous speaker's talk, the work I'll tell you about has to do with characterizing the physical and structural properties of cells. But as you'll see in my talk, we're focusing more on the proteins and their interactions with one another. So I'd like to tell you about the work that we're doing in my lab to develop and apply proteomics approaches for studying the architecture of macromolecular machines, as well as the architecture of gene regulatory networks. It's a really exciting time in the field of mass spectrometry-based proteomics, and part of the reason for the excitement is that the instrumentation that we're using just gets better and better, it seems like every year. So the machines are becoming faster, more sensitive, and we can acquire data that has higher and higher resolution. And this information combined with computational approaches is really allowing us to both identify and quantify proteins in a way that was really not possible just a few years ago. And in addition to that, we can combine these proteomics techniques with biochemical approaches to begin to identify protein-protein interactions on a much more rapid scale and more sensitively. And so I'll tell you about 3 projects that are going on in the lab today. The first one involves using proteomics approaches to characterize gene regulatory networks underlying red blood cell differentiation. The second topic will be our development of crosslinking and mass spectrometry approaches to study the architecture of large macromolecular complexes. And in the final part of the talk, I'll tell you about our efforts to develop mass spectrometry-based approaches to study protein-protein interactions on a large scale. And so the rationale for these studies, of course, is that we know that most cellular functions are carried out by large macromolecular assemblies. So if we want to understand how these cellular processes are regulated, we need to understand something about the complexes that are involved. So we need to understand their composition, of course. We need to understand how the composition changes as cells transit through different states. And ultimately, we want to know the architecture or structure of the complexes so we can better understand how they function to control these processes. And you can see that some of these macromolecular machines are quite complex, like the nuclear pore complex can have up to 456 proteins. So we really need powerful technologies to be able to address these issues. And the other aspect is that we know that information flow is controlled by molecular interaction networks, such as protein-protein interaction networks and gene regulatory networks. And we know that these networks changes in the architecture of these networks underlie all cell state transitions. So if we're talking about the transition from a normal cell state to a diseased cell state, or the differentiation process, there are changes in the architecture of these networks that underlie these cell state transitions. And it will be important for us to be able to decipher what the architecture and the changes in the architecture are of these complexes so we can understand better how these cell state transitions occur. Okay, so as I said, there's 3 topics for the talk today. The first one's going to focus on our development and application of targeted proteomics approaches to quantify transcription factor proteins during red blood cell differentiation and how we've used this information to develop dynamic network models of the transcriptional regulatory networks underlying this process. And so this is a collaborative project with 2 other groups. In my group, there's Mark Gillespie is a research scientist who did all of the mass spectrometry measurements. In Ottawa, we've collaborated with Marjorie Brand. She's an expert in red blood cell differentiation and transcriptional regulation. And another person at Ottawa Hospital Research Institute is Ted Perkins, and his student, Danielle Sanchez-Tataval. They are computational biologists, and they helped us with the dynamic network modeling. So as we know, red blood cells differentiate from stem cells through a pathway shown here, where we go from stem cells differentiating to common myeloid progenitors, which then differentiate to megakaryocyte erythroid progenitors, onto erythrocytes, and eventually the red blood cell. And we know that transcription factors play a key role in driving the transitions between these different cell states. So GATA1 and PU1 play important roles at this branch point, and KLF1 and FLI1 playing an important role at this MEP transition to the erythroid or the megakaryocyte lineages. And furthermore, it's thought that it's actually the stoichiometry of these factors that plays a critical role in driving these cell state transitions. So high levels of GATA1 at this CMP stage is thought to drive cells to the MEP stage, whereas high levels of PU1 is thought to drive the cells to the GMP stage. And similarly, at the MEP stage, high levels of KLF1 are thought to drive these cells to the erythroid lineage, whereas high levels of FLI1 would drive the cells to the megakaryocytic lineage. So to clarify the role of transcription factors such as these in this differentiation process, we thought it would be a good idea to actually measure their expression patterns throughout this time course of differentiation, and to do it at the absolute level. So we want to know the copy numbers of these transcription factors and how they change throughout this differentiation process. So the outline of the experiment is described here, where we are going to try to establish dynamic models of erythropoiesis that incorporates changes in the copy number of transcription factors across time. To do this, we're going to use an ex vivo model for erythropoiesis. This is a system that Marjorie Brand has developed in her lab, where we start with CD34 positive stem cells, and we can differentiate them to mature red blood cells in a process that apparently recapitulates what happens in vivo by exposing the cells to a variety of growth factors and by co-culture on a mesenchymal cell layer. Marjorie prepared the cells in this way and then isolated nuclei from these cells. She provided the nuclei, and we made nuclear extracts so that we could perform a type of mass spectrometry called selected reaction monitoring. I'll tell you a little bit more about this technique in the next few slides. So this will allow us to measure the absolute abundances of any proteins we'd like to throughout this time course. We're going to focus on transcription factors. We've also done RNA-seq on the same cells that we're analyzing by mass spectrometry. And then the protein level expression measurements and the RNA level expression measurements were used as the input parameters for the dynamic network modeling. And so selected reaction monitoring is a really fantastic mass spectrometry technique. And it's great because it's really sensitive and it's very reproducible, and we can do it in a quantitative fashion. In order to do selected reaction monitoring, first we have to develop assays for each protein that we're interested in. And to do that, we have to first identify so-called proteotypic peptides. These are peptides that will be able to uniquely identify our protein of interest in our biological matrix. So we go through databases and find these peptides. We selected 423 peptides corresponding to 167 transcription factors for the first round of this experiment. Then we need to synthesize heavy isotope labeled peptides corresponding to the proteotypic peptides. So this will tell us we combine these isotopically labeled peptides with our biological sample so that when we're running our mass spectrometry experiment, we can look for the signal due to the heavy isotope labeled peptide. And this tells us that that is the signal that corresponds to the peptide that we're interested, or the transcription factor that we're interested. It also allows us to determine the absolute quantity of the protein in that matrix, because we know exactly how much of this heavy isotopically labeled peptide we spiked into the sample in the beginning. And then we program the mass spectrometer. In this case, it's a triple quadrupole mass spectrometer, and it will monitor for the presence of that proteotypic peptide during the time course of this mass spectrometry analysis. And we can monitor for approximately 200 different peptides, or 300 different peptides corresponding to that many proteins or fewer in about an hour in one of these experiments. So we can really measure a lot of proteins and do it really accurately and determine the absolute levels of these proteins through this time course. So it's a really nice technique for doing systematic quantitative studies of proteins. And so this is the result after lots of work, lots of mass spectrometry runs. We were able to actually quantify the absolute abundance over the time course of 100 transcription factors and cofactors. And here you can see the clustering of these transcription factors. We were able to measure about 3 orders of magnitude of protein abundances for all these transcription factors, down to about 100 copies of a transcription factor per cell, up to about 100,000 copies per cell. So it's really a lot of nice quantitative information. One of the first things we did, of course, is we compared the RNA expression profiles to the protein-based expression profiles. And as you might expect, we see a reasonable correlation between these two measures of abundance. But there are certain proteins where the protein level expression measurements do not correlate so well with the RNA level expression measurements, such as this protein TAL1, which we know is an important transcription factor in this differentiation process. So this indicates that there's probably some post-transcriptional events going on that are regulating the expression of this protein, and this is important to know as we go forward and think about the underlying transcriptional regulatory mechanisms that are going on. Another thing that we looked at was this idea of the stoichiometry of transcription factors during the time course of differentiation. So these two transcription factors, KLF1 and FLI1, are thought to be cross-antagonistic. And one idea is that when their levels become equimolar, that indicates a cell state transition. In this case, we see that they become equimolar when you look at protein level measurements of their absolute abundances at the time point that corresponds with this MEP population. And this is the stage where they're actually thought to either drive cells to the megakaryocyte or erythroid lineage. But if you look at quantitative RNA measurements, you see that you would say that they become equimolar at a much earlier stage. So this is, again, an example where protein-based measurements might lead you to a different view of the underlying biology than the RNA-based measurements. So our ultimate goal at this point is to use this information to derive network models, dynamic network models of the transcriptional regulatory process. And so to start, we started with 15 core transcription factors that are known to be important for driving this process. And we considered proteins as the regulators and RNA as the targets of the regulators. And the quantitative changes in transcription factor levels and their absolute abundances were used to predict the behavior of possible target genes. So we can, for each of these transcription factors, we considered each one for their ability to regulate each of the target genes. And in this way, we were able to generate dynamic network models. And you can see here the results of this analysis, where we see our regulatory, transcription factor regulatory network throughout this time course, up to day 10 in this case, where blue lines indicate activation, red lines indicate repression. The transparency of the lines indicates the importance of the transcription factor for the regulation of its target. And the thickness of these lines is an indication of the strength of the transcription factor in regulating the gene. So because we have this absolute abundance measurement, these absolute abundance measurements, we can actually incorporate this information into these models, which is a really nice feature. So in this case, a thick line indicates that it's a strong activator of E2F4, is a strong actor of KLF3. So relatively few molecules of this protein were needed to drive expression of this target gene. So this is really exciting, I think. It's the first time that I know of where protein-based measurements and absolute abundance measurements have been used to model the transcriptional regulatory network underlying different red blood cell differentiation. There's a number of course, a number of known regulatory interactions in here, such as the GATA1, PU1 cross-antagonism, and the KLF1 and FLI1 cross-antagonism, and we can see the dynamics of the effects of these proteins on each other as we move through this time course. There's also, of course, a lot of previously unknown interactions, and we're interested in characterizing these in the future. To try to validate the model for now, Marjorie Brand's lab has done knockdown experiments for a few of these factors, in this case GATA2, GATA1, KLF1, and TAL1, and she has shown that the predicted interactions are borne out by the RT-PCR analysis for these factors. So it seems like these models are giving us useful information describing the regulatory network underlying this process. So this is going to be an iterative process, right? We're going to continue to make more measurements of transcription factors through this time course and try to incorporate them into the model. And we also, as we test the validity of the model through these knockdowns, we'll incorporate that information and refine the model. So for this part of the talk, to summarize, I told you that we've established a method to perform absolute quantification of transcription factors during the time course of erythropoiesis using selected reaction monitoring. We've quantified 100 transcription factors through this time course. Globally, there's a good correlation between the RNA and protein changes. However, we see some transcription factors that display a high degree of post-transcriptional regulation. And we use absolute protein abundance measurements to generate dynamic network models of erythropoiesis. We incorporated both the temporal abundance measurements as well as the absolute abundance measurements to estimate the strength and importance of each transcription factor in the regulation of their target genes. And some of the current ongoing studies are to compare accessible transcription factor binding sites with transcription factor bound sites with the absolute abundance measurements we have to see how these relate to each other. Okay, so that's the first part of the talk. Now I want to move on to our studies where we're trying to develop structural proteomic technologies to study the architecture of large macromolecular complexes. So there are many ways to study the architecture of protein complexes and protein-protein interactions, I'm sure you're aware. High-resolution techniques like X-ray crystallography and lower-resolution techniques like yeast 2 hybrid. We really like mass spectrometry combined with affinity purification and cross-linking because we can take advantage of the sensitivity, the sensitivity of the mass spectrometer, its ability to identify proteins in a relatively unbiased manner, and our ability to actually do the structural analysis on native complexes. That is, there's no need to crystallize the protein complex. We can do the analysis right on the proteins in solution, hopefully under physiological conditions. So in the end, it's really a nice complementary technique to all these other structural biology techniques, and it can give us really useful information, especially for some of these complexes that I'll tell you about that are really large and flexible, and they're quite refractory to techniques like X-ray crystallography. So cross-linking combined with mass spectrometry is a protocol that we and others have been using for now quite a while to study the architecture of complexes, and the technique is outlined here, where we start with a purified or partially purified protein complex. We expose it to a cross-linking reagent. In this case, I'm showing you the BS3 cross-linker, which reacts primarily with amine groups, so lysine side chains and the N-termini of proteins. And the idea is that the cross-linker will link residues that are in close proximity within the protein complex. And then if we can extract this information, that is identify the cross-linkages by mass spectrometry, in the end, we get information about the relative proximity of these residues within the protein complex. So after cross-linking occurs, we then digest the proteins into peptides. We typically use trypsin. Tryptic peptides fly well. They are good substrates for mass spectrometry analysis, so that's why we do that step. Cross-link peptides are typically quite low abundance in these complicated mixtures because the cross-linking reaction isn't very efficient. So we typically do some kind of fractionation or enrichment of the cross-links before going to mass spectrometry. Then we go on to the mass spectrometer. We do high-resolution LC-MS to characterize the cross-link peptides. And then we have to use some database search algorithms that can specifically identify cross-link peptides. These search algorithms are different than algorithms that are used to identify standard peptides when you're trying to do, say, just protein identification, but not cross-link peptide identification. And then if all goes well, we've got a really nice data set. We can identify hundreds of cross-link peptides often for a particular protein complex. And this gives us a lot of information about the organization of the protein complex, right? We know which proteins are close to each other within the complex, and not only that, which parts of the proteins are close to each other within the complex. And the lab has worked really hard over the years to develop this entire workflow for doing cross-linking. We've done this kind of analysis on probably 20 different macromolecular machines now. We're developing our own cross-linkers that have special properties. I'll tell you a little bit more about some of these at the end of the talk, hopefully, if I have time. We have worked on developing the algorithms that allow us to confidently identify the cross-link peptides and computational tools to visualize the cross-linked maps. So I'll tell you about one example of how we've applied this technology to study the architecture of a macromolecular machine. In this case, it will be this macromolecular complex called TF2H. So what I'm showing you here is the RNA polymerase II transcription machinery, which is required for expression of all protein coding genes in eukaryotic cells. And it's a complicated machine, as you can see. Over 60 polypeptides are required for expression of a protein coding gene. The process typically starts out with the recognition of the promoter by this complex called TF2D. It itself is a complicated machine composed of 13 or 14 polypeptides. So it first recognizes the promoter typically and recruits the other components of the machine to the promoter, including TF2H. And TF2H plays a really important role. It has a number of enzymatic activities that allow it to carry out its roles. And the roles that we know that it functions in is to separate the strands of DNA around the start site of transcription to allow the polymerase to gain access to the template strand so it can begin to synthesize RNA. And that activity is thought to be catalyzed by one of its enzymatic activities, this XPP ATP-dependent translocase. It has another important activity for transcription, that's to phosphorylate the RNA polymerase molecule on this part of the molecule called the C-terminal domain of the large subunit. And it's believed that phosphorylation of this tail subunit of polymerase is important for driving the complex from the open state to the elongation state. So it helps for the polymerase to break its contacts with the rest of the components of the machinery. And that activity is catalyzed by its kinase activity. So we want to know what the structure is of TF2H so we can understand better how it functions in this process. And when we started this work, we had these nice EM images of both the human and the yeast complexes. And this is really great. It gave us the overall shape of the complexes. You can see that their shapes are quite similar. So it's a highly conserved macromolecular complex through evolution. But we really don't know the organization, right? We can localize some of the subunits in these complexes, but we don't know the overall subunit organization for the 10 polypeptide subunits in this complex. So we performed the cross-linking analysis, and you can see the results summarized here. We're able to identify approximately 100 cross-links, both cross-links between subunits and within subunits. The cross-links within subunits are typically easier to capture because these regions are usually more fixed in the complex, and they're close together. But this gave us a lot of information about the overall subunit organization, and importantly, information about which parts of these different subunits are close to one another within the complex. And the combined results for both the yeast and the human are shown here, where you can see that we were able to identify over 35 conserved domain-domain interactions within this complex. So this is really nice information. It gives us a lot of information about the overall organization of the complex, and as I said before, about domains that are in close proximity within the complex. And right now, we're quite interested in understanding how the enzymatic activities of the complex are regulated. And so now, for example, this XPB subunit, we know that's the one that's important for separating the strands of DNA around the start site using its translocase activity. And so now we have information that this protein P52, and in particular its hub domain, is located in close proximity to a region of this translocase subunit. And so we're focusing on that to try to do mutagenesis studies to see whether or not it truly is required for regulating the activity of the translocase. We're going to do similar things with the kinase subunit now that we know that this latch region of the MAT1 protein is in close proximity to that kinase subunit. But we wanted to get a three-dimensional representation of the complex, and so in order to do this, we collaborated with the Andre Sali's group in UCSF, and they have this really fantastic computational approach that allows them to take structural information from diverse sources, in this case cross-linking information, cryo-EM data, as well as atomic level information for parts of the subunits. And through a computational approach, they can integrate this information and produce models that best satisfy all of this structural information as distance constraints. And shown here is the top scoring model from this analysis for both the yeast and the human TF2H. And you can see that their overall subunit organization is quite similar. Their shapes are a bit different, But now we know a lot about the overall organization in a 3D representation, and importantly, we know information about where particular domains within these subunits reside within the complex, and this helps us to design our mutagenesis studies to test predictions from this model. So we get a lot of information from this type of cross-linking mass spectrometry combined with computation approach. And so far I've told you about our work to study the structure of macromolecular complexes involved in transcription regulation, but we know that there are so many protein-protein interactions in cells. What about all the other interactions that we're interested in? So we know that there's protein-protein interactions drive so many biological processes. And I think that this approach, cross-linking combined with mass spectrometry, can provide a high throughput way for us to study protein-protein interactions in cells in a large-scale fashion, and to do it so that we can look to see how protein-protein interactions change over time or through a time course of differentiation. And I think we can do it because, of course, mass spectrometry is so sensitive and it's a high throughput technology. So if we can get cross-linking to work efficiently enough and get those cross-linked peptides to the mass spectrometer, we should be able to get a nice global view of protein-protein interactions in any cell at a particular time. It's going to be, of course, challenging because there's so many interactions to look for, right? So we know that there's thousands of proteins expressed in any cell. These proteins probably engage in tens of thousands of protein-protein interactions. There's a huge dynamic range of protein abundances, which is going to make identifying cross-links between lesser abundant proteins difficult, of course. Currently, our cross-linking chemistries we know are not very efficient, so we're going to have low abundance of these cross-links. And I didn't tell you about how we actually identify the cross-linked peptides, but typically we don't measure the masses of the two linked peptides, so we have to use computational approaches that can consider all possible combinations of peptides that could produce the fragmentation spectrum in the mass spectrometer. So we need ways to be able to confidently, that makes it hard to confidently identify a cross-linked peptide when you're dealing with large numbers of proteins. So we need to figure out ways to confidently identify cross-linked peptides by searching whole proteome databases. So to try to address these issues, both the identification and the confident identification and the detection of these peptides in mass spectrometers, we are developing new cross-linking reagents that have special features. One feature is they have enrichment handles, like a biotin handle, so we can actually fish out the cross-linker modified peptides from these mixtures that have many, many peptides in them. And they have these groups called an MS labile or cleavable group within the cross-linker. And what this allows us to do is to actually fragment the cross-linking reagent within the mass spectrometer so that we can actually determine the masses of the two cross-linked peptides. Once we have this information, then we can confidently identify most cross-linked peptides by searching a whole proteome database, so we won't have this issue of confidence in cross-link identification. And we've begun to do some of these studies by applying some of these cross-linkers to complex mixtures. In this case, we've applied this cross-linker called 2XL-DSB to a yeast whole cell extract. And while we didn't identify thousands of protein-protein interactions, like I hope we can do eventually, we did identify 100 cross-links between proteins, and these 100 cross-links corresponded to 67 protein-protein interactions. And as you might imagine, these interactions are between proteins that are highly expressed in cells, like ribosomal components. So it's promising, I do think, but we need to further develop the technology so that we can get to our goal of being able to reproducibly identify hundreds or thousands of protein-protein interactions. And I think the main issue is that the samples are just still too complex. So we are working on developing efficient and specific enrichment of these low abundance cross-linked peptides from complicated mixtures. And my hope is that we're going to be able to, in the next year or so, be able to perform routine and systematic analysis of large-scale protein-protein interactions for any cell type or organism. And we can do this in a dynamic fashion so that we can look to see how these protein-protein interaction networks are changing for example, as cells move through differentiation programs or cells go from a normal state to a disease state. I think having this type of information will give us a lot of insight into the molecular mechanisms underlying cell state transitions. So it'll be a nice complement to, say, our global RNA measurements and our proteomic measurements. Now we'll actually have interactomic measurements if we're successful. So that's all I have for you today. I told you about our efforts to develop targeted proteomics strategies to systematically measure transcription factor abundances during erythropoiesis, and how we use this information to generate transcriptional regulatory networks, how we're using cross-linking mass spectrometry combined with integrative modeling to study protein complex architecture, and our developments of cross-linking mass spectrometry technology to study large-scale protein-protein interaction networks. A lot of people contributed to this work. I want to acknowledge, in particular, Mark Gillespie, a research scientist in my lab who did all of the mass spectrometry work for the erythropoiesis project. And Jay Lowe is a senior research scientist in my lab who did all of the cross-linking mass spectrometry work that I described today. Our collaborators for the TFIIH project were Dylan Tages at University of Colorado, and Andrei Sali at UCSF, and Steve Hahn at the Fred Hutchinson Cancer Research Center. And Marjorie Brand and Ted Perkins were our collaborators on the erythropoiesis project.